Okay, so welcome back. Again, we're in the Master of Science in Bioethics at St. Thomas University, and this is the course on bioethical issues at the beginning of human life. And today we're going to talk about stem cell research and cloning. Okay, so we begin as always in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for your many blessings, for the gift of life, and for having us here today as we seek to delve deeply into these bioethical issues at the beginning of human life, trying to uphold and promote always the dignity and the sanctity of human life from conception to natural death. We ask for inspiration today as we look at these topics of stem cell research and cloning, that we may be able to see what is your truth about these issues and seek to promote it among those who surround us. As always, we pray in a special way for those who are in most need of your divine mercy, in the name of Christ our Lord. All right. First of all, any questions or comments from previous lectures or anything on the topic? No? Okay, we're going to move forward then. First with stem cell research, and then with cloning, human cloning, right? If we were to ask the question, what is your opinion? Do you think that the Catholic Church is for or against stem cell research? Just to frame it like that.
It's for stem cell research using the correct stem cell.
Right. Using the correct stem cell, right? So you make a qualifier. In other words, if we were to, and if I ask the general public and even the students here, the undergrads, you know, what's your opinion? What's your feeling that the Catholic Church is for or against stem cell research? And I just leave it at that. Say, oh no, they're against stem cell research. The Church is against stem cell research. And so it gives me the opportunity to say that not only is the Church not against stem cell research, the Church is actually doing stem cell research, okay? But obviously it's adult stem cells and not the embryonic. So we have to make that distinction. And forever it's making distinctions in bioethics. It's making a lot of distinctions to get to the very core, because when we get down to it, it's the way that the stem cells are obtained that is either ethical or not, okay? So many times it's about the means that are used. All right, so when we talk about stem cell research, we have to make distinctions at least at two levels. The first level is between human and non-human stem cells, all right? When you think about this, there are stem cells in just about any species in nature. I mean, when we talked about asexual reproduction, right? Asexual, excuse me for a moment. Let me stop the recording. I'm sorry, just a minute, please. So I was saying that we have to make the distinction between human and non-human stem cells. That's the first level of distinction, all right? If you recall when we were talking about reproduction, the two modes of reproduction, sexual and asexual. In asexual reproduction, stem cells are involved in the regeneration. For example, remember some animals, when they lose parts or pieces, they're able to regenerate it. Well, that's due to stem cells that they have in the body. We all have stem cells. And also even the cloning aspect that happens with vegetative propagation in plants and so forth, all right? So there are stem cells in nature. Just about any species will have stem cells. They typically are not a lot, but they're there, and they're embedded in different organs and tissues for regeneration and for repair, really, for cellular repair. At any rate, the first level of distinction with regards to stem cell, as far as the bioethical analysis is concerned, is whether they're human or not. Okay. And then the second level of distinction, of course, within the human, if they are embryonic or adult. And this is really the core of the bioethical distinction, if they're embryonic stem cells or adult stem cells. So we're talking about human First, we're going to talk about human embryonic stem cells and where do they come from. And you'll see right away, I hope that it's very intuitive once you see where the human embryonic stem cells come from, that it is unethical to participate in this kind of research. Okay, oops. All right, so human embryonic stem cells, abbreviated as HESC or HESC, right? Sometimes you only see SC, stem cell only, and they don't tell you what the origin is, all right? So sometimes in the literature, you just see SC, stem cells, so SC research, and it doesn't give us the source. But we need to know if they're embryonic or adult, because the embryonic stem cells, they come from the inner cell mass of the blastocyst. Remember, the blastocyst is the third stage of that embryonic development. Zygote is the fertilized egg, then goes to the morula, and after the morula, we have the blastocyst. In other words, when that little morula starts hollowing out on the inside and the cells begin to differentiate, the first level of differentiation, whereby you get an outer layer of cells called the trophoblast, and then this inner mass of cells inside the hollowed cavity, that's the ICM, the inner cell mass, okay? And remember also that the trophoblast will eventually become what? That's in closer contact to the endometrium, so it will become the placenta, right? Whereas the ICM becomes what? The embryo proper, right? That's what will develop into the gastrula, and then through the gastrulation process, the 3 germ layers, ectoderm, mesoderm, and endoderm, and then from there, the next step is to begin to differentiate the different organs and organ systems of the body. And so the ICM is truly the embryo proper, right, of that early embryo, right around the time of implantation. Well, it is precisely the ICM that researchers go after to make these embryonic stem cells. Those are the embryonic stem cells. So you see that functionally, what needs to happen is in the lab, take these blastocysts, of course, before implantation, so they're not going into women's wombs and scraping out blastocysts, okay? These blastocysts, where you think is their source of the blastocysts. They're not implanted, but the real life in vitro fertilization, the ones that are cryopreserved, the spare embryos. Okay? Remember, so this is a way that the industry, that the in vitro industry has figured out to get even more money for the procedures and at the same time get rid of those spare embryos because the parents don't want them, they have been abandoned or whatever, there are too many, okay? No woman is going to implant a dozen embryos normally, correct? And so they donate them to science. The in vitro clinics donate them to science. But of course, so the donation is for free, right? So it all stays in nonprofit and all this, but they're entitled to compensation for the time and effort that they have put into generating these blastocysts. So there is the back door opens for the flood of money to come in and to compensate them for the technique of obtaining them, of generating these blastocysts, the whole incubation process, right, at body temperature and all that, and then the cryopreservation for who knows how long a time, because those are just expenses of the industry, all right, and they're entitled to be compensated. Just like people in nonprofit organizations, they're entitled to salaries. That's compensation, that's not part of the profit. You know what I'm saying? And so that's how they're actually getting paid for these embryos. And in my opinion, it's just human trafficking. It's at the microscopic level, but it's very much, if the blastocyst is a human being, then that's human trafficking. So in order to, back to the stem cells, the researchers will take the ICM, which means that they have to disaggregate the blastocyst. They have to tease it apart, all right, with sophisticated instrumentation, of course, micro manipulators and all that under the microscope in the higher standard of quality control and antiseptic environment and all that. It's put into a Petri dish, and then that ICM is further desegregated and is, and the hope is that this ICM, these stem cells will then be encouraged to form any one of the cell lines that make up the human body. And we have about 220 cell lines in the body, distinct cell lines, that are, if you will, the precursor of all the organs and tissues that we have. Okay, so there would be, for example, a number of liver cell lines, neuronal cell lines, any tissue in the body, pancreas, blood, bone, muscle, you name it, those are cell lines, about 200 of them. And they are the precursors to all the differentiated tissues and organs that make up our body. Okay, so why do they go after the ICM? Why is the ICM theoretically the gold standard for stem cells? They are the biological gold standard, but I emphasize that this is theoretically, all right? Well, of course, in the process of obtaining the ICM, the embryo is killed, right? And then they use euphemisms like destroyed, but it's not really destroyed because we destroy things. It's kind of, to use destroy is consistent with a utilitarian mentality, by the way, because we destroy things, you know, we destroy the phone, we destroy the chair, whatever, but those are objects. So these human beings are not really destroyed. Human beings are killed or left to live. Right? But you see, the euphemism is consistent with that utilitarian mentality of seeing the unborn as an it, as a thing, as an object. All right, so why is the ICM the gold standard for stem cells? Exactly, they're totipotent. They're totipotent, okay? Actually, take it back. Not really totipotent, they're pluripotent, because you see there's already one level of differentiation. The the morula and the zygote are totipotent, all right? But by the time you get to the blastocyst, because there's one level of differentiation already, which is the trophoblast, all right? They're actually pluripotent, which is better, because pluripotent means that the ICM will become the embryo proper. In other words, all of the organs and tissues will come from the ICM, right? Except the placenta, which for the born, we don't need placenta, okay? Because that vital organ has done its function already. So they're actually pluripotent, and pluripotent is the first level of differentiation, if you will. So they have a lot of potency, to the point that the ICM, the inner cell mass, will generate all of the organs and tissues in the body. So you can see that theoretically they are the gold standard, because in principle one could get all of the 200 cell lines from the ICM, right? Since the three germ layers, ectoderm, mesoderm, and endoderm, the three germ layers that form the organs, they come from the ICM, right? The three germ layers, they come from the ICM. So we can see theoretically that yes, they are the gold standard. There's two more steps of differentiation just to complete the picture here. This is with regards to cell potency and what we call differentiation, cellular differentiation. See, all the organs and tissues of our body are differentiated. What does that mean? That they do specific functions and not other functions. The heart doesn't think, and the stomach doesn't pump blood, and so forth. You know, each organ and tissue has a particular function. Together, they form the organism that is living. If everything is metabolizing the way it's supposed to, then we have a living organism. And this is consistent for every organism. Any multicellular organism will have differentiation, cellular differentiation. Another way of saying differentiation is specialization, specialized organs and tissues that do their particular function, okay? All right, so that's the standard. And again, we have about a dozen organ systems in the body, depending on how they're classified. All right. What is a system? What are organ systems in the body? Circulatory, nervous, digestive, gastrointestinal. And what puts it all together, what holds us together, literally, physically, physiologically, is the integumentary system. Integumentary system is another, just a fancy word for saying the skin that has several layers, because the skin involves the epidermis and then the dermis. Well, first it involves a layer of dead skin that is surrounding us, that is actually there for protection. Then there is the epidermis, the dermis, the conjunctive tissue, and on all the little glands that are inserted in the dermis. For example, the hair follicles, the nerve endings, etc., sweat glands, and so forth. So really, the skin system, the integumentary system, is an entire system. Okay, it's composed of organs that have similar function. At any rate, I say it doesn't more or less depending on how they classify, because you can look at the muscular system separately and you can look at the skeletal system, but there are also sometimes functionally they look at the musculoskeletal system together. And that's why the number is a fuzzy number of how many organ systems we actually have in the body. All right, anyway, it's a little too much detail, but just to point out that the aim and the goal of growth and development, organically speaking, is cellular differentiation into the distinct organs and tissues that we have so that we can function normally, physiologically, like we do, all right? That differentiation is much more drastic during the embryonic development, right? Remember, I think I mentioned one time I did a little bit of the math of how much growth we have just physically from the zygote to birth, and then from birth to death, right? Assuming a normal life of about 80 years, from the zygote, which is pretty much the size of an egg, of a human egg, to birth 9 months later. So the diameter of an egg is about 10 microns to a foot more or less of a baby on average. If you do the math, it's about 5,000 times larger, all right? So we grow physically 5,000 times in 9 months, less than 1 year, which is extremely drastic. Just to have a contrast of the ratio, we grow 5,000 times during pregnancy, right, for 9 months. The rest of our lives, once we're born, if the baby is 1 foot, let's say we grow to 6 feet, which is a tall person, right? Most people are not 6 feet, but just for the ease of math. How much more have we grown the rest of our lives from birth to death? From 1 foot to 6 feet, that's 6 more times only. So compare 5,000 to 6. In other words, we do the bulk of our growth, we do embryonically in the womb, protected. That's precisely the whole purpose. Functionally, the purpose of pregnancy is to protect that embryo during that extremely fast, neck-breaking speed development that occurs in the embryo. And to boot, it's even shorter than that when you consider that by the 2nd month, right, by the 8th week, all of the major organs and systems are in place, but in miniature, because that little embryo is about 1 inch long. All right, so really, the most drastic growth that we do and differentiation is in the first 2 months of our entire life, first 2 months. And the rest is mostly growth in size. And so from 1 inch to 1 foot, we grow 12 times bigger, 12 inches in a foot. And that's mostly just growth in size of the organs, except for a few organ systems. One that is non-functional during the entire pregnancy because it's not needed, right? Because the embryo is being fed, or the fetus is being fed and cleansed metabolically through the placenta. And that's going to be the pulmonary system, right? So the respiratory system, at the level of the organs of the lungs are non-functional until we're born. So that's one exception where the organs are still non-functional, even though they're there intact. Then the other one is the system that takes the longest to develop, right, which is going to be the nervous system. And there, the neurologists are telling us that the synapses, the not complete integration and the full network of synapses of the brain, especially the cortex, the cerebral cortex, which with all its convolutions, which is where thought occurs, the higher thought, the abstract thought, which is the highest process that we have available, that takes about 20 to 25 years, okay? And so that is really, that's when we can talk about a neurological adult that is capable, on average, of doing abstract thought, the highest capacity of the brain. So you can see that the nervous system is very, very slow growing indeed. Okay. Then, for example, you can look at the reproductive system. That takes about 10 years after birth, on average, to be functional, and so on and so forth. But this has to do with differentiation, one way or another. Okay. So we have these 4 stages from totipotent, typically a single cell or a small cluster of cells that can become any cell of the body. And that's also why I was making the argument, the blastomeres, right? What's a blastomere? Remember? Blastomere is an individual cell of the morula, right? It's called blastomere. These individual cells on the morula, they're also totipotent. So that's one of the issues against pre-implantation diagnosis, right? If we pluck one of these out, who can tell us that we're not generating a new human being? Because now we have a totipotent cell that is isolated from the rest of the cluster. So we have compromised its integrity. The rest of the cluster remaining inside the zona and the corona have their own integrity, so that can continue to be an individual. And imagine, and these cells are so plastic, in other words, they are truly so totipotent that if you pluck 1 or 2 out, that morula, on average, can continue normal development. All right? So that's pretty interesting. At any rate, by the time we get to the beginning of differentiation, of cellular differentiation, then it loses some of the potency to pluripotent. What does this mean functionally? It means that the ICM is not going to generate a placenta. And it also means that the trophoblast will not become the embryo proper, will not differentiate into the 3 germ layers. Okay, so normally differentiation is a one-way street. This is important for when we talk about cloning. In nature, differentiation is always one way, from less differentiation to more differentiation. That's in nature, it's always that one-way street, okay? When we go further into organ systems, that's when we get into multipotent. Multipotent are stem cells that are already toned down into a particular organ system and not another one. So we can talk about circulatory stem cells, or we can talk about, let's say, muscular stem cells or bone stem cells and so forth. They are already semi-differentiated or further differentiated into particular organ systems. And they're called multipotent, right? They're not fully differentiated yet, but they are, if you will, the most immediate precursors of the fully differentiated cells. Okay, so it's a step further differentiation. I'm emphasizing this because when we'll see with adult stem cells, multipotent cells are actually better than pluripotent cells. You'll see why. Okay, but anyway, so the embryonic stem cells, they come from the ICM, which is pluripotent, and because all of the organ systems are going to form from there, theoretically, they are the gold standard. They're supposed to be the best stem cells. In practice, okay, so what happens in practice? When these experiments have been done in animals, okay, because to do it in humans, you need the permission, the FDA permission for clinical trials and all that. There have been some attempts, but the protocols were rescinded because before they got they saw the writing on the wall that they were going to get sued uh, royally. So I'll talk about that. But when this is done in animal models, and what would be the best model in which to do this, again, functionally, if we're looking toward the healing potential of stem cells for humans? The standard model, who's going to get it? In the lab, the mouse. The mouse is going to get it, right? If they're small, we can breed them fast. Each litter drops 4, 6, 8, 10 mice, and it's about every 3 weeks. So just about any lab that is doing stem cell research or any kind of genetics or genomic research, they're going to have labs, they're going to have breeding quarters for the mice, all right? And some of these are transgenic mice. So there are a number of mice that are available today. You can even buy them, and you can put in your specifications of what kind of transgenic mouse you want, and some labs will do it and send you the mating pair so you can start your colony and so forth. Anyway, the whole point here would be, what would be a protocol to test for healing? All right, so we take, it's always the female that gets it, right? So we take a female mouse who the eggs are extracted. Obviously, we need to go in vitro because we need a blastocyst that is not implanted, correct? So the female mouse will be hyperovulated, she'll be extracted, functionally just kind of the same technique, only smaller. And now we're going to have a dozen eggs or whatever on the Petri dish. Then the male mouse, the sperm is obtained, the fertilization is done in vitro, the incubation and so forth, according to the mouse standards, whatever incubation time that is, probably just a few days. And eventually we get to the mouse blastocyst. All right, so now we got mice blastocyst. The researcher will go in, take out the ICM, put it on the Petri dish, and grow it to what is called confluence. Confluence is when the Petri dish, the cells tend to grow laterally in a layer, in a thin layer of cells, right? Until they reach the edge of the Petri dish. If they're not transferred to another Petri dish, they'll continue to grow and overgrow on top of each other. So when they reach confluence, you have to do what is called passage to other Petri dishes and so forth. That should keep the cell lines going. And in principle, they become immortalized. In other words, they continue to regenerate themselves and grow, as long as the proper conditions are mimicked. In other words, the nutrient medium of the agar will have mimics the environment, the nutrient environment of the uterus of the mouse in 37 degree, being a warm-blooded animal and so forth, okay? Then what's gonna happen? These ICM then we take, so we want compatibility. The first issue with the ICM is that it's going to be these embryonic stem cells where they're transplanted into the patient, right? Which in this case would be a mouse. The first thing that happens is that the patient's body rejects those stem cells because they're not fully compatible. So it seems like a foreign, it's like a transplant, it seems like a foreign object. What do? They go back to the female who, from where they extracted the eggs, right? And they injure that female. The typical injury is going to be an injury to the spinal cord to see if they can regenerate the neuronal stem cells, which are the neuronal cells are the highest specialized, the most specialized, and the ones who regenerate the least. Right? They regenerate the least, and so they're trying to get the neuronal stem cells to act, the stem cells to regenerate neuronal tissue. So what they'll do with that female is they'll injure the spinal cord and sever, but not total, they'll do a partial severance of the spinal cord, and they inject the stem cells into the area of insult, in the area of damage. They'll inject directly the ICM, right? In the hope that what? That these embryonic stem cells will then differentiate and generate neuronal stem cells and bridge the gap. That's the thing. All right. So two things happen functionally in the lab when this is done in practice like this. The first thing is that the mouse's body rejects those stem cells because they're only 50% compatible, right? In other words, the egg did come from the mother, but the sperm came from the father who is not the same identical genome as the mother. And so there's a rejection there. That can be overcome because functionally it's similar to a transplant. A transplant, by definition, is not from the same patient, right? But it's from another human being. So it's the same species, but different individual. So how do they deal with transplants? How do they tone down the rejection?
They give them medication.
Exactly, immunosuppressors. So it has to do with the immune system. What is it that attacks that foreign body that seems like an infection, right? It's the immune system. It's the immune system. So there are immunosuppressors that tone down the immune system. They don't shut down the immune system all the way. It's not an all or none thing, because if you shut down the immune system totally, then that mouse or the human, in the case of transplants, is susceptible to all kinds of infections, bacteria, fungi, and spores that we're breathing in and out right now in the air, because the air is not sterile, okay, and life is not sterile. And therefore, it's just toned down. It means that these patients are more susceptible to infections. So for them, a cold could transfer into a pneumonia and stuff like that. They just have to be more careful, more prudent. Normally, they shouldn't be in rooms with a lot of people, close quarters. Airplane is a problem. Traveling by air is a problem because of the recirculation of the air. So they have to wear masks, so on and so forth. Okay, but the ways of getting around that. The greater issue is this. Because these cells are pluripotent, meaning that they can become any organ or tissue of the body, they actually grow too fast and don't really differentiate into neuronal cells necessarily. They end up becoming tumors, cancerous tumors, which is definition of a cancer is uncontrolled cellular growth. So, bottom line, when embryonic stem cell research has been done in the lab in models, mammalian models, typically the mouse, sometimes they use the rabbit, whatever. The biggest issue is that this ICM, these embryonic stem cells, are causing tumors. So again, translate that into the human. Here you have a human being who is injured for some reason, and he gets, the embryonic stem cells. Now, in addition to the injury and the disease that they have, they're also going to grow tumors, cancerous tumors. It's a bad situation. We don't want that, okay? And so, to this date, the researchers have not been able to cause this embryonic stem cells to not grow tumors, all right? And that's the challenge there, which, thanks be to God, it's a beautiful thing because just on the science of it, the researchers are walking away from these embryonic stem cells, which means that fewer embryos are being killed for that, right? And so basically is that the ICM has too much potential. It has too much potential. And I just make a simple little analogy. If we had a radio when we used to have transistor radios and run by battery, right? So I have this little battery, but instead of being the normal, what are they, 9 volts or whatever, 12 volt battery, it's a 200 volt battery, okay? But it's the same size. So when I put that into the radio is going to fry because it's just too much potential. And that's what's happening here. Functionally, they have too much potential. They grow uncontrollably. And it's like the whole embryo wants to grow in that side of injuries. So theoretically, they're the gold standard, but in practice, they're absolutely disastrous. For years, researchers were empecinados, were stubborn on trying to get this thing working, and they couldn't. Finally, I think it was Geron. Geron was one of the first biotechs that came up on the horizon around the 70s maybe, or 80s. I think in the 70s, when these biotechs started in California, because California a few years ago had an amendment to their constitution where they carved out, I think it was $8 billion for stem cell research, for embryonic stem cell research, through the state university system of California. And they established a stem cell research institute in La Jolla, California, to get this free because they wanted to be the precursors. We're talking about 10 to 15 years ago, more or less, at the beginning of the 2000s. Anyway, it's an old company, biotech out there, I think it's called Geron, and they wanted to start a trial. After they had done many, many trials and tweaked it, and they thought they had a model ready for human embryonic stem cells, but they weren't going after something as drastic as spinal cord injury or something like that. They were doing something with macular regeneration, trying to reattach the cornea or something. So they were going after something that is not life-threatening for the individual because blind people can still live, right? So in other words, the eyes are very important organs, but they're not vital organs. So they're ready, and they even got FDA approval for the protocol for the human trials, okay? And as they started the human trials, they noticed they were starting to have problems with the volunteers for the trials, okay? It wasn't quite working as they were supposed to. And so right away, they rescinded their protocol and they gave up the license that the FDA had given them to do that research, to do the human trials to this day. And that happened about 2-3 years ago or something like that. After years, because the embryonic stem cell research has been around for decades at this point, all right? And they still can't get it to work. We say thanks be to God, because they're walking away from the embryos now simply on the clinical side, on the biological side, that they don't work. So it's still a utilitarian mentality. They're not using them, not for any consideration of the embryos, but simply because they don't work. However, because some unscrupulous scientists want to continue, they're again stubborn about this gold standard thing, some labs have gone after the ICM and continued the embryonic stem cell research. Because why? Oh, they're growing uncontrollably. That sounds like cancer. Let's use them for cancer models. And now let's try, see if we can get this ICM to be coaxed into the different cancer types, because it's not cancer, it's cancers, all right? Depending on what the tissue is and the organ has been affected and so forth. So now some labs and some researchers are using the human ICMs for cancer research. Okay, it's not as many, so the number is down, but there is still to this time, and that was the big thing with, so Bush W. Jr. signed we call it a line in the sand, a decree or executive order, whereby no federal monies would be used for generating new human embryonic stem cell lines. Wisconsin had developed a number of lines that were about, throughout the world, there were maybe 60 or 70 lines that had been developed, human lines that had been developed from embryonic stem cells. And because it was not illegal, and they were even receiving federal funds for that and so forth. So Bush Jr., I remember, because I was in Boston at that time preparing for graduate school, he, by executive order, forbade federal funds for that kind of research, okay? Which means that state funds are available. That's why California went the state route, and private funds are available, and it's not illegal. But of course, we know that the two biggest generators of research in the United States are NSF and NIH, right? So NSF is for the non-human experiment. Their budget is, I looked it up last year, for NIH, I think it was $13 or $14 billion annual budget that's for the clinical, for the human research. So National Institutes of Health, NIH, that's the federal branch that is in charge of medical research in the US. And then NSF, National Science Foundation, they're in charge of sponsoring non-human research, right? Those are the two big branches in the United States, and it's to the tune of billions of dollars annually. So these are the two big engines of research scientific research in the United States, which then gets published throughout the world, and it moves the rest of the research in the rest of the world and so forth. All right, so it became illegal to use federal funds, but state funds and private funds are available, and it's not illegal, so therefore it's the technological imperative, right? Anything that can be done will be done sooner or later. So that's kind of the history of embryonic stem cell research. We're opposed to it in principle because it kills human beings. Basta. If we were able to obtain, individual cells from the blastomere, from the ICM, and not damage or hurt that blastocyst significantly, then we would be in favor of it in principle, because it would be a good thing for humanity. Some very positive research could come out of that, like curing diabetes and so forth. All right. But when you think about it again, how are we going to obtain just a few cells from the ICM without compromising the integrity of that blastocyst? It's extremely risky, and there's no guarantee that fully, because at this point, because there is that first level of differentiation, what is to tell us that if we pluck out just even a few cells, that is not going to be maybe the ectoderm layer or the mesoderm or the endoderm is going to interfere with gastrulation? Because at this point, we're already talking about the embryo, purely embryo cells. Number one. Number two, again, we're not going to go into women's womb and be pulling out blastocysts as they seek to implant. So we would have to collaborate with the in vitro industry. We would have to acquiesce to the whole illegality, the immorality of manufacturing human embryos in the lab for the purpose of research. So really, bioethically, there's no way around it, all right? However, the good thing is that adult stem cells are actually working. So we all have adult stem cells in our bodies, and these are ASCs. Again, sometimes they just, the researchers and the papers, you only see SC research, stem cell research. I think it's unethical not to tell us the origin of those cells. You have to get into the materials and methods, and you have to really dig deep into what were the sources of the stem cells. If they were adult or embryonic, sometimes you have to go into the supplementary data that is only maintained online and so forth. So anyway, the A stands for adult, and so that's something very positive, meaning that we all have stem cells in our body, okay? There are stem cells, for example, at the follicle, the hair follicle. We pull out a hair, sooner or later that hair is going to come back, right? Well, because the hair follicle has stem cells that will generate a new hair. What else? We got stem cells all over the place. The liver, you can cut out about 1/3 of the liver will regenerate. It's amazing. Got a lot of stem cells. It has a lot of regeneration power, okay? Even in the hippocampus, they've discovered stem cells, which this is really a very interesting discovery, because the hippocampus is kind of in the center of the brain, deep inside, and apparently oxygen stimulates. There's some neuronal stem cells there, and oxygen stimulation will get these stem cells to differentiate into neuronal cells wherever they're needed in the nervous system. Mm-hmm. There are very few, but they have been discovered. And apparently, one stimulant for them is oxygen. So there are stem cells around. There's stem cell in the adipose tissue. Another word for adipose is just a fat tissue. So when the people get the liposuction, you can get the stem cells out of the liposuction. Okay.
Nobody wants those to regenerate.
Yeah. That's right. About for the skinny ones like us. Yeah, so now they've discovered stem cells even in the blood system, which means that a simple process, relatively simple process, or a standard protocol, like which is analogous to pheresis, for example. I don't know if you've ever done pheresis, but that's for obtaining platelets. Now stem cells can be filtered out of the blood. So the procedure is similar to pheresis. They lay you down on the bed, they put some blankets because you're going to get cold, and then they take out the blood from one arm, filter it, and spin it, and pump it back through the other arm. And then it takes several hours, but they are able to obtain a few dozen stem cells from the patient with a minimally invasive procedure. Oh, bone marrow is stem cells. And it wasn't really known as that, but bone marrow transplant is one of the earliest transplants that was done precisely for the hematopoietic stem cells, the stem cells that are coaxed into their precursors of the RBCs or the white blood cells, right? And platelets. So bone marrow has been classically a source of adult stem cells, typically from the iliac, is that the iliac region, the bend of the, yes, the iliac crest, is it? Right, of the hip. That's where, because there's a large source of bone marrow there, okay? Anyway, so there are sources of stem cells. The pulp of baby teeth, for example, when we shed our teeth, that has stem cells in it also, et cetera, et cetera. So there are sources of stem cells, adult stem cells in our bodies. Again, they're not a lot, they're few, but they're there, okay? And they're just waiting to be stimulated into differentiation whenever it's needed. So for example, if I cut myself, that cut is gonna regenerate from the inside out. And that regeneration is coming from stem cells that is producing new dermis and epidermis and so forth. So there you have it. Now, they're not pluripotent, they're multipotent because the vast majority of our organ systems are differentiated, right? So they're already in particular organ systems. And they reside in that organ system and normally don't move around, right? So multipotent is better because that means that their potency is toned down and they're not prone to making cancer cells, turning into cancer cells, okay? So they're toned down and that's a better deal. Now the small battery is beginning to look more like a 9-volt battery or 12-volt, whatever it has to be, okay? So toned down. Also, the other advantage is that we can obtain stem cells from the patient himself or herself, and therefore there's no immune rejection because the tissue is compatible with self. And those are called autologous transplants. Autologous coming from the same person, the same patient, okay? And there's compatibility with self. It's essentially, it comes down to extracting cells from one part of the body and injecting them into another part of the body. Like, for example, the knees for the regeneration of the meniscus and so forth, or the cartilage. It's been done, and it's quite successful. At various stages of development, they have, the researchers have been able to obtain results, and there is this web page that is called Stem Cell Research, and since the 2000s, for about 20 years now, they have been documenting all the scientific literature on the success of adult stem cells. Stem cell research, just one word. These are ads. See, there are ads already, and you see regeneration. Now there's a lot of quackery here too, so you have to be careful. Oh, I guess I need to put the .org. See, this field has advanced so much. It used to be able just to put stem cell research and their site would come up. There it is. But now, because there have been so many successes in this area, you have to put the .org into it. Otherwise, you get a lot of publicity and then also a lot of research. I saw NIH there, one of the sites of stem cell research success. So this is their site. This is a group called Do No Harm. And this group was put together, like I said, about 20 years ago now. And what they do, they document, they're constantly scouring the literature, the scientific literature, and pulling out all of the articles, or retrieving and saving all of the articles that have some success, some level of success with adult stem cells. And even embryonic stem cells, if they could find it, but to the date, they're 0. So let's see if they still have it. Down here at the bottom, they have a little, like a clock. Yeah, there it is. So far, they have identified 73 successes at different levels of the trial with adult stem cells and 0 on the embryonic. It's interesting. And so at some point, I think it's this one. Yeah, okay. So you see, adult stem cells over here, embryonic stem cells. And these are scientific articles, plugging anywhere. Let's see. This is where it was. Well, this is just the list is on that site. There's a place. It's a place where one could link in here, click, and see the actual articles. But anyway, this started, like I said, about 20 years ago.
Okay.
Yep.
I have a question. When we say adult, we are referring that they are not embryonic or
Exactly. They come from the born.
exactly.
So, right, good
There's
questions.
no age for
Exactly. So let's look at that again functionally, how that happens. Adult, we would have to put it actually in quotations if we were rigorous, because adult is from the born forward. All right. Now, even a baby has adult stem cells that are available. Now, what's the crucial issue here, bioethically speaking? Of course, consent, informed consent. It's so obvious that we don't think about it, but we have to obtain consent from whoever is donating the stem cells, even the patient. We would assume that the patient is going to agree to donate to self, right? Because they have the advantage of the potential for healing, but that informed consent has to be in writing and has to be informed, so the clinicians have to tell the patient what's going on and so forth. And then sign on the bottom line. Who signs for minors? The parents sign for minors. And that's legal and ethical, right? Because it's for their good and it doesn't really damage them. The most that the damage is some kind of a puncture wound to extract the stem cells, but that would be done in the proper clinical setting with sterilization and everything else. All right? So that's the good thing about adult stem cells. They're working. Now, the bad thing is that they seem to be kind of temporary. They don't stick around. And this is what I've heard mostly. One of the ones that have been most successful is with some arthritis of the knee or the degeneration of the cartilage, right, the cushion. That stem cells have worked there, but after a while, after a few years, they wear out. And so the person needs like a reboosting. The patient needs reboosting of another treatment, another round of stem cells. Okay, but again, they would be autologous, adult autologous stem cells. So at least from the bioethical perspective, it's still ethical. The problem is they don't seem to do the full recovery yet. But so that's what they're working on at this point. But you can see, excuse me, just by putting stem cells in here, you get so many ads and hits. Right? So example, where it says ad, it's an ad. Okay, Stem Cell Miami. This is a .com here in Miami that is doing the process. And it's not illegal, so they're doing it. But see, hematopoietic, these are for blood related to blood or circulation. Mm-hmm. These are all ads, look at that. So it's working. I guess the FDA has given them approval. They're adult stem cells, and that's a good thing. It's looking like adipose tissue.
Are they referring to plasma rich for their injection or different procedures?
No, it's they're spun down and purified, okay, and isolated.
What they do in the offices is just plasma rich.
Yes.
Where they, you know, like you described the
Yeah.
procedure, that's what they inject in the joints.
Right. So that could be.
I don't know if that's what they're calling stem cell.
Probably. So the short answer is I don't really know the details of the clinical aspect, but I'm thinking that the plasma rich probably has stem cells in it, right? Circulatory stem cells, and that's what they're looking for. Mm-hmm. So it's even more benign in the sense I imagine they don't have to go through the whole phoresis process, right? They just take it's a regular donation of blood and then just spin it down.
Yeah, so that is what is popular. You know, a lot
is
of
it?
orthopedic offices or pathologists.
You see? Yeah. So it's even less invasive. It's less of a hassle for the typically it's autologous, right? It's a self donation.
Yeah,
it's your own blood.
Same,
right.
They take it out, they spin it down, and then they
inject it in the site, directly into the site. Yeah. Okay. So yeah, if they get result with it, then that's a positive thing. The thing is, so I think that in general, blood donations are always separated here in the United States, right? I think that's the standard. When donating blood, they will separate it automatically. And that's typically why one can eat, and actually the blood banks want donors to have eaten well before donating. So their blood is strong and so forth. I went, when I was in Italy, studying there and so forth, one time I went to donate. So at that time I was CMV positive, negative, which means that blood qualifies for infant transfusion. CMV is that virus, cytomegalovirus, that is innocuous to the adult but can be damaging to the babies. And so when one is CMV negative, that blood can be distributed into the little quad packs, the little smaller containers or bags for infant transfusion, for baby transfusion, right? And so I went to the infant hospital there. They were asking for donations, in particular thing. I'm O negative also, which is a universal donor. So the vampires are always after me for that. And I went to donate, and on purpose, I ate a nice large breakfast and everything else. And I got there all excited, and it came up in the conversation that I had just eaten. It says, "Oh no, we cannot accept it. You have to be fasting." It's totally, they do whole blood. And that's the standard in Europe, to the point that they didn't even tell me when I was making the appointment and all that, you have to be fasting. But that's the standard over there. It caught me by surprise that it's a different protocol for handling the blood. Isn't that amazing? Anyway, that's stuff behind the scenes. Yeah, sure.
Of course, it has to be your own stem cells, right? Because there's a lot of publicity that I have heard. For example, when parents, they try to preserve the stem cells of the umbilical cord.
Right.
And they say to parents, no, you can use it even to the baby, the siblings of the babies, to the parents. But I mean, that cells in theory will only work for the baby, right?
Yes, but that's 2 slides down. To the umbilical cord stem cells, because there's a particular category that is very promising. And so I'm going to get to that in a couple of slides. Okay, so umbilical cord stem cells, yeah. Hold it there for a moment. All right, so this is just a general area of adult stem cells. Let's look at a couple others, which are the induced pluripotent stem cells, the iPSCs. And then the next one is going to be the umbilical cord, all right? So this fellow, Yamanaka, this was in the early 2000s, because I was, at that point I was at Purdue doing my research. And since the lab where I was working, it was doing structure and function of DNA, we were all interested. And it was the big talk at that time in the labs was the potential of the stem cells and all that. It was very big at that time. So this fellow Yamanaka is the one who was able to do this induced Shinya Yamanaka. Induced pluripotent stem cells. Oh my goodness. Yeah. Induced pluripotent stem cells. This is how it works. Yamanaka is from Japan, and all the different labs in the world at that time, at the beginning of 2000s, were competing with each other to see who could get those embryonic stem cells into cell lines, okay? And so it's a lot of competition going on. And Yamanaka was doing embryonic stem cell, human embryonic stem cell. He was getting blastocysts from a local IVF clinic, or who knows how, but he was getting human blastocysts, disaggregating them, plucking out the ICM, putting them in the Petri dishes, incubating, getting confluence, and trying to coax them into the 200 cell lines. They figured that if they get about 100 cell lines, that would be the precursors for all of the organ and tissues of the body eventually. The idea was to develop an international library of stem cell lines that would be available for clinical trials throughout the world. That was the whole idea. So different labs of the world were collaborating and competing at the same time, right? Because there's also in the background to this, potential Nobel Prize, et cetera, publications, you name it. All right, prestige, research. So Yamanaka was disaggregating these blastocysts, human blastocysts under the scope. And at one point, he thought, and I know because I was reading his declaration when he was interviewed, he said, these blastocysts look very much like my daughters looked like when they were at that stage of embryonic stage, okay? So here's a man who was honest with himself. He had daughters, I think it was 2 daughters, and he knew that his daughters looked exactly like that when they were blastocysts, okay? So I said, there's gotta be a way around this, rather than killing these human embryos, okay? And so he started working with mesenchymal cells, mesenchymal all from precursors of muscle tissue, fibroblasts, human fibroblasts. They're the precursors of the muscle tissue. We know that muscle eventually differentiates into 3 types of muscle: skeletal muscle, smooth muscle, and cardiac muscle, right? Cardiac muscle is the heart. It's the only cardiac muscle. And the characteristic that it has is that it's self-beating. It has its own pulse, in addition to the innervation of the autonomic nervous system. Then skeletal muscle is connected to bone and innerved by the voluntary nervous system, so we can contract skeletal muscle. Thus, we're able to move the skeleton, literally. But smooth muscle is not connected to bone and is not innerved by the voluntary, it's innerved by the involuntary nervous system. But it is innerved, meaning that it will function through control and coordination of the autonomic nervous system, which will be the autonomic functions of the body, like, for example, digestion and every other function that occurs automatically, even the breathing that occurs at night, for example, right? That's all done by smooth muscle. Anyway, these are the fully differentiated tissues, okay? The precursors to these are these fibroblasts that are part of the mesoderm in the germ layers. So you can take it all the way back to the gastrula, right? But these fibroblasts, because they generate the muscle tissue, they are multipotent. They're not pluripotent, okay? Which we know is a good thing, functioning. All right, so what Yamanaka did was he started experimenting with these fibroblasts to see if he can get them to undifferentiate back to pluripotent, but not to totipotent. So take them back only to pluripotent, to the previous potency, right? So these are multipotent. To take them back to pluripotent, but not totipotent. Because he knew that if we would take it back to totipotent, then we're back to the same thing. We got a zygote, and therefore the potential of a new human being and so forth. So he wanted essentially to generate something equivalent to an ICM, but just on the potency part, not an integrated whole like a blastocyst, which is a human being, all right? So we wanted to get really the functional equivalent of an ICM, but just at the level of the potency of the cells. All right, so it turns out that it has to do with developmental factors. In other words, these factors typically are proteins in a cell, and that growth that I've talked about from a single cell to the 5,000 level growth, the differentiation, even just in the first 2 months, how is that controlled? How is that coordinated? Again, there's no magic or anything like that, okay? Rather, what there is pathways of differentiation. And those pathways of differentiation, those are regulatory genes that are early on, the regulatory genes early on at the zygote, at the morula, at the blastocyst stage. What is coordinating the blastomeres of the morula to differentiate into trophoblast or ICM, right? Well, those are regulatory genes. The genes code for regulatory proteins. And the proteins are the ones that coordinate and control the pathways of differentiation. Here, we're getting down to the molecular level inside the cell. But basically, it comes down to proteins, regulatory proteins. The regulatory proteins are the ones that regulate differentiation, cellular differentiation. The ones that control, coordinate, right? These are regulatory proteins. And they occur early on in development. They do their function, and then they're put aside, they're digested away, and they're done their function and moves to the next level of differentiation and so forth. So he was researching with these proteins, and he was able to discover through a lot of hard work in the lab, hours and hours and hours of hard work, okay? Very little sleep with this whole team because there was a lot of pressure by other labs who were on it, that it came down to about 3 or 4 regulatory proteins. 3 or 4 regulatory proteins. In fact, one of them is Cas protein, which is also involved in the CRISPR-Cas thing of taking a DNA sequence of one species and inserting it into a specific region of another species, targeted gene transplants, exactly. But anyway, these regulatory, there are 3 or 4, came down to those 3 or 4, whereby if those 3 or 4 proteins are extracted, right, that cell, that multipotent cell will revert, will undifferentiate, will de-differentiate. It's called de-differentiation. It will undifferentiate. It will go backwards and become more potent. And so he took multipotent cells, took out the regulatory proteins, and those multipotent cells reverted to pluripotency. Okay? But that's why it's called induced, because this in nature just doesn't happen. It's going on I-95 against the traffic in reverse, okay? So reverse gear, step on the throttle, and just head back where you came from in full traffic. That's what this is, and it doesn't happen in nature. So they're induced, they can be done in the lab, but he got pluripotent cells, which are the gold standard, right? Okay. But what I found interesting from the bioethical perspective is that the motivation was a motivation of conscience, that he was honest enough to realize that he was killing human beings by obtaining that, by extracting that ICM. All right, so he made these induced pluripotent cells, which are available. For that, he got the Nobel Prize. well deserved and so forth. But there's a challenge. So the way he got these proteins inactivated was actually to silence the coding region for the protein. Okay, and to silence the coding region, he had to insert anti-coding regions and so forth. He had to really manipulate big time the genome of the cell. And to do that, he had to insert, so how do you insert genes into individual cells? You have to use some kind of a needle, some kind of a syringe. The syringe are viral capsules. Viral capsules. So you put the gene of interest in a viral capsule and you send the viral infection into the cell. And what goes into the cell is not the viral DNA, but the gene of interest. Okay, but of course, this is not done at the individual viral level, right? This is a shotgun thing. You try to get the viral purified to 99.9999% because you're dealing with millions or maybe billions of viruses in a single aliquot. And so it's a shotgun thing at the end of the day, which means that some of those viruses, maybe 0.0000001% of the viruses have the viral DNA, which could contaminate the fibroblast. And now you got a viral little cancer going in there as part of that whole lot of cells. So it's not 100%. And so there was complaint about that. There was also complaint that these regulatory proteins may have other functions in the further differentiation. So once these are undifferentiated to pluripotent, can then they move forward into a further differentiation, a secondary differentiation into the actual cell lines that they're looking for. And that wasn't 100% either. So there's a lot of back and forth to it. It's not really clean, but at least it's an attempt. And some labs are still sticking to it and trying to work it out. And so again, research, publications, grants, and so forth. So it keeps the oil keeps greasing the machinery. All right. But I mention it because they're still viable. iPSCs are viable, meaning research-wise. Okay. I'm not aware of any clinical trials and any human clinical trials, but there may be some animal trials with iPS cells.
But where was he getting those stem cells?
The blastocysts or okay. So the first time around, the blastocysts came from in vitro clinics in Japan. Or he may have even been generating, I don't know exactly. There was another fellow who was doing human cloning, Yang Hui, and he's in China. And he was just getting the eggs from the female researchers in his labs. The postdocs and the graduate students, he was just putting pressure on them to donate eggs because we're going to get the Nobel Prize for this. So the sources, it depends on the person. Now, when he switched to fibroblasts, human fibroblasts, and are available just from donations from patients with informed consent, because that's just a syringe, right? Okay. Further, the last one on the stem cell research scenario are the umbilical cord, all right? So the full name is the umbilical cord stem cells. They're also known just as cord cells, just cord cells for short, all right? It's a reference to the umbilical cord. And even that is kind of misnomer because it's not the actual cells that make up the umbilical cord itself, but is the blood that's inside the cord, which is a little blood. It's a little bit of blood, okay? But that blood has stem cells in it. Now, those stem cells are very nice because they're early on, they're pristine, they're tender, they have not been exposed to a lot of UV or anything like an adult stem cell has, right, over the years because they're from the newborn. And so they're young, they're stem cells that are young. Typically, a baby has a lot of regeneration power, potential. the tissues are still very young and pristine, and they can regenerate well. So they have all those qualities. In addition, they have, these are the mesenchymal, by the way, mesenchymal stem cells, or MSCs. In addition, they're bioethical. Why? Normally, in a birth, the umbilical cord becomes medical waste. It's going to go to the incinerator, right? Medical waste. Because the baby is now born, doesn't need the placenta, doesn't need the umbilical cord, and that's going to be discarded. So instead of discarding it, the nurse, after the baby is born and after the cord is cut, will insert a needle into the cord and extract the umbilical blood. Okay? Then that's sent to the lab, spun down, and the cells are isolated and cryopreserved for years. So, ethical or not ethical? Certainly, the baby is not harmed, and normally this would be discarded. The only thing that's required is consent, informed consent from whom? The parents, which means that they have to pay because it's not covered by insurance yet. Okay, not covered by insurance, so they have to pay. They can get the kit online. They can order the kits online and just tell beforehand to their doctor and to the nurse, we want to save the cord blood. We want to save the cord blood stem cells. Okay, so fine. Then the nurse has the kit and everything, and we'll go for it and do it. Then that's sent to a lab, and it's cryopreserved, and they're stored, so they have to pay for the maintenance, right? Which may be $200-300 a year. Over the years, it may add up. What's the hope? The hope is that they're never used. But if Junior eventually has an accident or an injury or an illness or something, well, she or he has the stem cells reserved there. Maybe they can be used, all right? We don't know exactly how long, 20, 30, 50 years. We don't know. They're cryopreserved. They're supposed to be in a state of suspended animation. So maybe they work. It's a risk. But in principle, they're good. They're positive. In addition to Junior, for which these stem cells are fully compatible because they come from his or her blood, right? Siblings are semi-compatible because they come from the same parents, all right? So siblings are semi-compatible with that. So it could be that maybe not this Junior, but the other Junior sibling got injured or sick. Maybe those stem cells would be good there. And even also for the parents, because the parents are 50% compatible. The mother is 50% compatible on the side of her egg genome. The father is 50% compatible on the side of his sperm genome. Okay, so the family, the immediate family could benefit from it. And again, if you think of it functionally, who is more likely to need them eventually more than the child would be the parents, actually, as they get older. But they're the ones paying for it anyway, so I might as well get something out of it. You know, so it makes sense. My argument with all this was insurance doesn't cover it yet. And so I'm telling couples, do it, you know, walk the line of paying for it, but then try to get your insurance to at least cover some of it for this reason. You would think that the insurance would be in favor of this because in principle, this has potential for healing and for less invasive procedures and so forth. So it could cut down the cost of medicine. It could cut down the cost of insurance paying for injuries and illnesses. So the insurance could eventually save something on their policies by promoting umbilical cord stem cells. See? They're definitely not controversial as the embryonic ones. They're not even as invasive as the adult ones to obtain them. And so I think there's a lot of advantage there. It's just that insurance today still doesn't cover it. Maybe they don't have the volume of it yet. They have to see the numbers to justify it somehow, right? But I'm thinking it's kind of a dog chasing its own tail. I think that if we get couples to save more and more of these stem cells and eventually get a national pool where they could be also karyotyped and whatnot and profiled just like we do with transplants, right? There's a natural registry of transplants. We could do a national registry of cord blood stem cells and then put it out there to see who can benefit from it. I think there is someone asking a question on the chat. Yes, Brazil. Oh, see, multipotent better than embryonic. Yeah, I think we answered that one. My husband works for a company that does this, so it's part of his benefits. Wow. Okay. Okay. Super. Right. So you see, yeah, this is a little message from Giselle. All right. So you see the whole point, right? Yeah. And I'll tell you another anecdote. When I was in, okay, sorry. So to answer your question first, the previous one, I think we answered that. Multipotent, better than embryonic, yeah, because the embryonic are pluripotent, too much potential, right? But the multipotent are already into a particular cell line or a group of cell lines. So they're belong to particular organ systems, and that's why, yes, they're better. You know that from your biology background. I'll tell you a little anecdote that happened to me when I was in Boston preparing to go into graduate school for genetics over there in Purdue. So I spent a year in Boston doing some research. I took some courses at Harvard and Boston University also to come up to date with what was happening because it had been 30 years since my bachelor's. And I also did some research. I was doing some research at St. Elizabeth Medical Center. St. Elizabeth Medical Center is a teaching hospital for Tufts University, which is also in Boston, and it's the medical school. The medical school of Tufts University is downtown Boston, and St. Elizabeth Medical Center is the Catholic hospital of the Archdiocese of Boston. Big, huge hospital. And on the side of the hospital is a whole separate building, about 4 or 5 stories, and that was the research unit of the hospital. Of course, they did all the testing of the hospital and everything else, especially the blood work and all that, but they're also doing research. So I volunteered to do some research there, also to get it on my package of application, right, and to get a letter of recommendation and all that. And in that hospital, a couple came to see me and they were concerned because they were having a baby and they wanted to save the cord blood and all this. And they had gone to a nurse at St. Elizabeth Medical Center, meaning a Catholic hospital, all right, in a big archdiocese, which is a teaching hospital for medical school. The nurse told the couple, no, sorry, we don't do that here because we're a Catholic hospital. In other words, the confusion was even at the level of a nurse in a teaching hospital. We should have known that if she had just worked it out for a couple of minutes, the cord blood, obviously they're not taking it out of the baby, okay? It's medical waste. That same nurse is putting that into a hazardous bag, biohazard bag for waste, you know? And the reason why the nurse told the couple they don't save the cord blood is because this is a Catholic hospital. But sadly, that nurse was misinformed. Later, I checked with administration, and yes, of course, they would save it. Okay, but this was about 15 years ago. Yeah, about 2001, 2001, more or less, almost 20 years. Anyway, I'm hoping that by today, more couples know about this and more hospitals know about this, that it's a very viable thing. And it turns out that Giselle's husband is actually involved in this kind of research. That's great. Monica?
I know my OB actually talked to us ahead of time.
Oh, yes, yeah.
So we were prepared.
Right.
And then we did that.
You did it, actually? Yeah, you still have it?
Every
Wow.
year, I think it's like $100.
Oh, okay, that's better,
I mean,
yeah.
it was more upfront,
Okay, yes,
obviously.
right,
It
yeah.
cost a lot upfront.
Exactly, yes. It could be several $1,000s, I think, maybe 1,000 or 2,000 at the beginning, right? To get them isolated and purified and all that. Okay, so it's in a storage, it's in cryopreserve somewhere?
Cryocell is a company.
There you go. You see? Exactly. So it's commercialized. They found a commercial for it. And yeah,
But I
super.
think it was good that we knew up front. You know, we had the information, we
Yes.
could read it, we could think about it,
Exactly.
and then the cost of it.
Right. Exactly. That's a big consideration. Yeah. There was no insurance kicking in here, right? You see? Yeah. So hopefully get the insurance industry to go this route. I'm thinking that it behooves the insurance industry. They can save some money in the long run. Yeah. Yes. Emma?
There's a two vessel cord. Can that be used for stem cells?
I'm not familiar with two vessel. What are you
That
talking about?
only has one artery and one vein.
Oh, yeah. Right. It's,
SW?
so I don't. Is this arterial blood or venal blood? Is that the question?
No, I'm just wondering if they can use the placenta.
Oh,
for
as a matter of fact,
stem cells.
yeah, they're also using the placenta, but I don't know where that research is at this point. But yes, the placenta has potential for, there are multipotent cells, or there would be pluripotent, but you see, maybe they're already coaxed into being only placenta. Mhm. I've heard that placenta is used in some countries to better the skin or something like that, right? To make it cosmetic? Yeah, okay. Oh, yes, I see. Okay, yeah. So they use it as a base, maybe as the collagen base or something like that, the organic base. But I don't think much as stem cell itself, as regeneration. Okay. Yeah, all right. So from here, I think we go into cloning. So I think this is a good moment to stop. Yeah. All right. So you have any questions or comments? Otherwise, I think it's a good time to take a break now. Yeah. Okay. Let's pause this. All right. So it'll take about 15 minutes, more or less. All right. Part 2. And we're going to go into cloning, human cloning. Again, a little bit of this, some of it is historical, but then it cropped up again last month with He Huankui. Didn't hear about He Huankui? Okay, tell about him, Jian Kui. We'll get to him. Let's look at cloning and human cloning and what's going on. So there are two types of cloning. This, again, is looking at it functionally. What is the intention of the cloning? It can either be reproductive cloning or therapeutic cloning, okay? But you notice there are two words, and one is the noun and the other one is the adjective. So the noun is telling us the what, the whatness, the substance, the substantive. We're doing cloning, all right? And the adjective, reproductive or therapeutic. In other words, it qualifies what kind of cloning or for what purpose. For the purpose of reproduction, for the purpose of generating a new baby to this couple who cannot have it, etc., or for the purpose of healing. In other words, essentially to generate blastocysts without having to go through the hypervalvulation and all that, and just get the ICM through a cloning procedure. Okay? Just giving you the big picture first. Basically, both are cloning, but one, the reproductive, is to generate a baby, to manufacture a baby, and the other one is to obtain embryonic stem cells for healing, for therapy. Okay, so now let's get a little bit on the background. At first, the language, because language matters, words matter, and the scientific way of saying cloning is somatic cell nuclear transfer. What that phrase does is it tells us how the cloning is done. What is the mechanism of cloning? Somatic cell nuclear transfer, or SCNT, or just NT, nuclear transfer. All right? So let's look at the biology of this for a moment. Somatic cell is a diploid cell, right? A somatic cell is a cell of the body. Soma, body. Soma is body. And so a somatic cell is every cell in the body except the gametes. So every cell in the body except the gametes is a somatic cell. It's a body cell. Those body cells, by definition, they are diploid, 2N, so they have a full complement of chromosomes. And in that 2N genome of every cell in our body, what do we have? How many pairs of chromosomes in a skin cell? 23 pairs, right? How many pairs of chromosomes in a hair follicle cell? 23 pairs. In the liver, in the brain, whatever cell except the gametes, 23 pairs. So they're diploid. Again, what does that mean? That means that each cell in our body has the full genome of the trillions of cells that we have in our body, has a full genome. So really the miracle is that we're not a mess, that we're not a chaotic mess, because if we have a full genome in every single cell in our body, thanks be to God that those cells are differentiated so that the vast majority of that genome is shut down and doesn't express. So that only the liver genes will express in the liver tissue and only the brain genes will express in the brain and the neurons and so forth. But the full genome is contained in every single cell of our body. And that's the whole issue about the supercompaction. Remember the 10,000 fold that the genome is shut down and only expresses a little tiny bit for that particular cell line, whatever tissue that is. Okay. But the point is that every nucleus of our body, right, has a full genome. Except for the gametes that are haploid, they don't have the pair. They don't have the pair. So these somatic nuclei, are in fact a package, a capsule of our entire identity, of our entire genome. And so this somatic cell nuclear transfer is a nuclear swap. It's a nuclear swap. We take an egg, we take an ovum, enucleate the ovum, take out the egg, take out the nucleus. We have an ovum, take out the nucleus of the ovum, which is haploid, and swap it, insert the nucleus of a somatic cell. We have done a nuclear swap, and what we have taken out is a haploid nucleus and replaced it with a diploid nucleus with the full complement of genome, and then leave it alone. Because the enucleated cell is an ovum, meaning that it has the regulatory proteins ready, set, go for, if there is fertilization, for dividing within 24 hours and eventually generating a morula and eventually generating a blastocyst and so forth. The regulatory proteins are in the cytoplasm of the ovum, ready to do their thing, to start development, okay? And so, in effect, what happens is that that nuclear swap makes the ovum now a zygote, functionally a zygote, but without sperm. Makes the ovum a zygote without sperm. There's no fertilization because the nucleus that the ovum inherited is a diploid nucleus. So there's no need for fertilization. Essentially, we can say that the biology, the theoretical biology of cloning or somatic cell nuclear transfer is generating a zygote of the same species without fertilization. Okay? Okay. So that can be done in the lab, and that was done in the lab, and the first clone mammal was Dolly, the sheep. Okay? And that's where it comes from. Now, you can see, for example, here is the link in Wikipedia, right? It's called somatic cell nuclear transfer. They're talking about cloning. And sometimes they don't even put the somatic cell, they just say nuclear transfer, which is really what it is. It's a nuclear swap. All right, so let's look at the first clone mammal. Here is the mother of Dolly, She's an ewe, right, which is a female sheep. And this is Ian Wilmot, Dr. Wilmot, and there is Dolly, right? This is now the model. This is the taxidermist result of Dolly. I'll tell you why in a minute. Wilmot and his team in the Roslin Institute in Scotland, right, cloned the first mammal in 1996 by nuclear swap. In fact, it was even a little easier than that. Instead of taking out the donor nucleus, the diploid nucleus, he just took the whole cell, the whole donor cell, and injected the donor cell inside the egg. Getting a little bit ahead of myself. Here is, in fact, this is not quite right. The cell came from the udder of the same female, so as to have genetic compatibility. So this female, this line should go all the way down here to the udder of the female. Anyway, picture this: a female sheep, right? Extract eggs by haploovulation. So we got now the egg. Then take a cell from the udder. They chose the udder, which is the mammary gland of the sheep. All right. I guess they were looking for tender cells, something, but they chose cells from the udder, from the mammary glands of the sheep, which are diploid cells or somatic cells, and inserted. So then from the egg, extract the nucleus of the egg, extract the haploid nucleus, and insert this diploid nucleus, the actual skin cell. They inserted the actual whole cell in there. That's all right, no problem, with the diploid nucleus. That's a nuclear swap. All right, this nucleus is enucleated, is taken out, and here is a cell inserted. And now this is a fused cell. It has diploid nucleus. This is in fact zygote sheep, same genome, right? A clone, and then goes to the morula and the blastocyst. When it reaches the blastocyst, they reimplant that little embryo into the same sheep who was a female, all right? She was ready for implantation because she had received the fertility drugs anyway to hyperovulate, and she was ready for an implantation, so there it goes. And then a few months later, I don't know how long the whatchamacallit is, the pregnancy for a sheep, maybe 2-3 months, whatever, maybe a little longer. Out comes Dolly. What is not correct about this diagram is that instead of having 3 sheep, it's just 1 sheep.
Okay,
It's
so
just 1 sheep.
the egg, the donor cell.
You have the egg coming from the ovary.
Is
It's the same sheep. You have a female, so you have an egg source, right? So the egg comes from the ovary of the sheep. The the diploid nucleus comes from the skin of the same sheep, and the embryo is implanted into the same female sheep. All right, so we're trying to do as much compatibility as possible and as least rejection as possible. And it worked. What many people don't know is that it took about 250 trials to work. I think 258 trials. Okay, so technically that's 250 abortions, right? To get it to one to finally work. Typically the embryos died, either the egg was not, the swap did not work, did not progress, the zygote did not progress to the embryo, to the morula and the blastocyst, or if they got it to the blastocyst, it did not implant, and they keep doing it over and over again until finally one finally did implant, and then survived the whole gestation period and died, and was born, sorry, and that was Dolly. Okay? Okay. And at this point also, it's might as well finish. And for that, I think he got the Nobel Prize too, the first clone mammal. This appeared in the second half of the 1990s. I was a pastor at St. Kevin Parish here in Miami. And that's when I saw this on the radar. I said, wow, So cloning a sheep in the lab and cloning a human in the lab is the same technique, because we're talking about a mammal. It's the same technique. It's a nuclear swap, all right? So biologically, in the lab, it's the same technique. It might not be long before someone wants to do a human cloning. And that's when I asked the bishop if he would send me for further studies. Ended up at Purdue, came back later with a genetics degree. But this is what the final thing, because it had been advanced. Stuff that was science fiction years ago was becoming a reality. Just like transplants for science fiction in the 30s and the 40s was becoming a reality in the 60s. All right. All right. So on the human, is the same. We have reproductive cloning or therapeutic cloning. Okay, before we get into that, just to complete, since then, there have been many cloned mammals. The next one that was done was the cat. And I remember it was called copycat, was the clone. The mouse was done. Many animals have been cloned. The dog was very difficult to clone, apparently, because I guess the female cycle is very tough. It's very hard to tell, to pinpoint when a female dog is actually ovulating, apparently, and they couldn't get eggs. They couldn't get, mature eggs. So it was, for some reason, it was very difficult to clone the dog. Yes.
The cat, have you seen the cats that have no hair?
Oh, yes.
Do you use those in the lab, or does it have to be a different cat?
Sometimes, actually, so they're nude cats and they're nude mice. They're nude mice, and they are for some experiments, yes, they're preferred because they're less liable to infection and to bacterial growth on the hair. Hair is extremely dirty at the microscopic level. Okay, it's like a zoo, like a whole forest, a zoo of bacteria and spores and viruses in the hair. All right, and so these nude mice were developed. It's a special breed for experiments, or maybe the cat. But I also know that there are nude cats. Yes, the Machisquinkle is the Mexican is from the time of the Aztecs. The Machisquinkle, yeah, they look ugly. They're just the pink skin, and the skin is wrinkled.
Yeah, they're like
It's something that came out of the ground somewhere. It looks like, they're special breeds. Anyway, so that's how Dolly was done, and the reason why they named him Dolly, because why Dolly, right? Because where did the diploid nucleus come from? So this sheep is a sheep because she has a full complement of chromosomes, and the full complement of chromosomes came from the diploid nucleus, right? And the diploid nucleus came from the udder. So it's an indirect reference to Dolly Parton. And her and her large breasts. Yeah, it's just tongue in cheek. I guess Wilmot and his team were fond of Dolly Parton and her movies and whatever, and it's just a reference to the other. Okay, so it's kind of ambiguous, it's disrespectful, but that's what it is, and that's the name Dolly. He said it. He said. And at one point he said it was because of her prodigious something. He said, yeah.
What she had to say about that.
Exactly. Yeah, I happen to be very fond of Dolly Parton because I think she's a great actress and a very moral and ethical person, by the way. But that's how it happened. Anyway, that's the fame and infamy of the sheep. I'll tell you how Dolly ended, the sheep, not the singer, in a minute, but let's look then what this is translated into the human model of reproductive and therapeutic, because this was actually discussed lively at the beginning of this century. All right. The reproductive cloning would be for a woman who, for whatever reason, could not get pregnant. Maybe his sperm, maybe he doesn't have the sperm count, maybe they're non-functional, incompetent, etc., etc. So she could end up with an egg and put the somatic nucleus of the husband. And that way it's kind of a quasi-fertilization, right? Because she's contributing the egg and he's contributing the somatic nucleus. Now we got the diploid egg. which technically now is a zygote. And then, so one of these would be a man and the other one would be a woman. They're both wearing, well, let's say this, no, that's it. Egg, sperm cell from a person to be cloned. If we were a married couple, for example, okay, because reproductive is procuring a baby for them. So let's say it's a married couple. The diploid nucleus, somatic cell, could come from the husband. And then we get the zygote, morula, implantation, 9 months later, baby on hand.
But in this case, father, they share the genes, right? In theory.
Right, well, it's the same species.
Because in the other case, it was the exact individual.
It's the same, yes, exactly, right.
The same sheep.
Exactly, because, yeah.
The same.
Yeah, and even with that was 250 trials. And then when they claim the mouse and the cat, and it's done now in animal husbandry a lot, it's done with cattle and so forth, because when they have a vigorous animal, they want to preserve that. But it's taking an average of about 200 to 250 in general for the mammals. So it seems to be at the mammalian level. That trial number seems to be at the mammalian level in general. So we could expect, based on that, for it to be the same for human. So it means that for every clone baby, there would be an average of 200 that died, okay, just to get that one. So it's a gross manipulation of human life. Again, it's manufacturing a baby in the lab. Necessarily also it has to risk the attempted implantations and all that. So it's a nefarious scenario. It's a gross manipulation of human life, and we should never go there. It's really literally manufacturing a baby in the lab. Number one. Number two, there is really no genetic mix. In other words, there is no fertilization. There's no sexual reproduction. So we're denying that child the benefit of the mix. Precisely, the benefit of sexual reproduction is that 50% of the genome of our genome, each one of us, came from our mother, and the other 50% came from our father. So we got the best stuff from our mother, genetically speaking, and the best stuff of our father, genetically speaking, right? So that if they were recessive genes, they would be masked by the dominance and so forth. All that is rejected because there is no fertilization, there is no sperm. So we are denying that child to be the best, the most vigorous child that he or she could be. We're denying the natural selection that occurs in fertilization. Okay, the advantage of the heterozygosity, the variance.
But because in this case, the somatic cell is from the father, then
right.
He's the one that is given all the genetics.
Exactly, 100%. So even in, exactly, it's a shell, and it's the regulatory proteins that would make the process go forward from zygote, but there's no genome of the mother. You see?
So if it's the mother, it's the wife of the man.
Right.
It's the same if it's a surrogate.
Exactly. Functionally, it's the same. She's a gestational mother, but she's not the genetic mother of that baby. Okay.
So the egg is like the case. Basically.
Exactly. Yeah, it's the encasement for the child, right? But so she's only the gestational mother of the baby, but she's not the genetic mother. It's the father who's 100% genetic. Okay, so really this child is a clone. It's a clone of whoever, of the father in this case, or if it's the same mother, so you have all kinds of bizarre things that can happen. You can go from the least to have only one. So for example, if they used the same diploid nucleus of the mother, all right, then she is the gestational mother, and she's also the genetic mother 100%. But she's also the sister. She's the twin sister of her daughter. Of course, the kid necessarily have to be a daughter because she's coming from the female genome. So simultaneously, she's the mother and the sister, but they'll be maybe 20 years apart or something like that. And they're identical.
They will be the same.
It will be identical.
Exactly, physiological. Exactly, like an identical monozygotic twin.
With 30 years
of difference, chronology, right? And it's bizarre. Because that kid, when that kid grows, are you my mother or are you my sister? Okay, and what's going on here? So, and where's my father? No need. Well, the Y chromosome is disappearing anyway, right? So might as well start early. The other extreme is you can have about 3 or 4 parents, because you can have the donor egg. You can have a donor egg from one woman, a donor somatic cell from another man or woman. That's 2 people. The third one who gestates, and the fourth one who is paying for the whole procedure.
Right.
And it's just getting baby without contributing anything but the dollars. So you see, it just lends itself to an incredible manipulation. We cannot assume that everyone else out there has the same principles and values that we have. So if it can be done, the technological imperative. It's not illegal. It's not illegal. So what drives it? The economy. I just want to do this. This is fashionable. Wow, this is exciting. This is interesting. Think of the entertainment industry and stars and actors and so forth. They have a wonderful body. Well, pass it on, right? Let's get another Shirley Temple going 30 years later.
or parents preserving to get another child if the other one dies.
Yeah, which is beginning to be done now in the pet industry. When the pet dies, they can clone it. It's expensive, but they can clone the pet now. Yeah. Mm-hmm. Yep. That's the industry. You know, there's $1 to be made. It's not illegal. It's just expensive. And it's trial and error. So when we take it to the human, it's a different story. Reproductive, on the other hand, that was reproductive cloning, okay? Therapeutic cloning. Essentially, the idea is to come up with blastocysts that are not the product of fertilization. All right? In other words, all we need to do is get the women to donate their eggs, and then we get the somatic cells, and we do the nuclear swap, and we generate a zygote, then we incubate the zygote to the blastocyst stage, and by the time we get to the blastocyst stage, we extract the ICM and put it in the Petri dish, and now we got our pluripotent cells without any fertilization. So we're not killing anybody. That's their claim. Of course, it's a bogus claim, because we're saying that once that egg became a zygote, we could assume that God has donated a soul, because that could develop into a morula, blastocyst, implant. 9 months later, a baby is born. That baby is not going to be soulless. So we have to trace it back to the earliest possible moment when the baby got the soul was at the nuclear swap. If God can foresee if that baby was going to survive or not. Okay. So if that baby was on a survival trajectory, we can assume that God would donate a soul. So we're talking about a human being back again, and this is essentially manufacturing human embryos in the lab without fertilization, just by cloning, to obtain the stem cells. All right. So even though this is not doing an injustice to a new baby who is simultaneously the twin and the son or daughter of whoever donated the somatic cell and all that mess, you know, there's no child involved here. There's no baby involved. This is just to obtain the stem cells. In a sense, this is even worse, because this is manufacturing human embryos in the lab solely for the purpose of obtaining their stem cells. Exactly. Their humanity. So it's a production line of manufacturing, literally manufacturing human embryos in the lab so as to obtain their stem cells. Okay, so to me, this is even more offensive, the therapeutic cloning. And yet it's not illegal.
It's because for them an embryo is tissue.
Mm-hmm.
That's the same.
Exactly. And that it has a soul, well, that's your opinion. Because,
But for me,
exactly, that's the challenge. All right, so you have seen then that these are all scenarios. These are the 2 types of cloning for the human, therapeutic or reproductive. Thanks be to God, they're not working well. They're not giving results, again, because there was an outrage here. Oh, back to Dolly. The scientific community at large eventually spoke against reproductive cloning. It was surprising and a happy surprise. And it was based, again, but it was based on a pragmatic utilitarian thing. It turns out that Dolly, the sheep, she died, actually, she became old prematurely. Yeah, premature senility. She started developing arthritis when she was still young, and other symptoms. She would start banging into the walls, and some symptoms parallel to dementia. So the conclusion is that she became senile prematurely as a youth.
He was taking a somatic age cell already. It's an adult cell.
Exactly.
So she only has certain time left.
Yeah, exactly. So the development was truncated,
Exactly.
and for humane reasons, she was euthanized.
Oh, wow.
She was killed because she was suffering, or she was in pain anyway. We don't know if she was suffering, but she was in pain. And so she was killed, she was euthanized for humanitarian reasons. And that seems to be the case with mammals. The mammals have that been, they mature fast and they become senile prematurely before the time for that species in general, okay? But because she was so famous, she was taxidermed, she was preserved, and this is the taxiderm Dolly. And that's why she's so still there in the photograph, okay? Both hairy, but this is the taxiderm Dolly as far as I know. It's they did the photograph after. But she did go to adulthood, and she did breed, all right? So that was proof of principle, just like Louise Brown, all right? The proof of principle was to get the clone to reproductive maturity and to actually have ewes or whatever they're called, kids, the babies, and then get those babies to grow and have the babies are normal.
But she didn't reproduce.
Yes, I think so.
Oh, she did?
Yeah. Yeah, because that was the proof of the. Now comes the doubt. Let me punch it in and see.
I thought that she died. I didn't know that she.
I think she did. I think she was mated.
Not often.
Dolly lived her entire life. Rossi. Yeah, she was bred. She was bred.
Produced 6 lambs.
6 lambs.
Wow.
Yeah. Right. She was still younger here. Right. So, and I think that the ewes or the lambs were also fertile. Okay, so that would have been, and I think that they were normal, her offspring, which that's the proof of the pudding, right? That's the full proof, what we call the proof of principle, that the cloning did work for, at least for the next generation. But she did age prematurely and was euthanized. So based on that, the scientific community at large spoke against reproductive cloning. That would be inhumane to do this to a human being because of the possibilities that that human being would be defective with age prematurely and develop these characteristics early on that are typical of elderly people. However, and so now the scientific community says, no, we're against cloning. And we're back to that deceptive language of saying, is stem cell research good or bad, right? We're against cloning. What they're against is reproductive cloning. But we say, no, the other one, therapeutic cloning, that's not cloning. That's somatic cell nuclear transfer. That's just nuclear transfer. This is cloning, meaning reproductive cloning, but the other one is just nuclear transfer, meaning the therapeutic. Because in the nuclear transfer, there's no baby involved. Sure, there's no born baby, but there's a human embryo. And that's the part they don't want to admit, because like you say, it's just tissue, right? And it's legal anyway. So this is what's going now, and you'll see it in the literature. In fact, to get that, that money through the legislator, through the state legislator of, no, it was a ballot amendment. So they put it in the ballot for the whole people of California. okay? And the premise on the document to try to convince the people what was put out to the public so that they would vote in favor of this amendment was um, that they were against human cloning, referring to reproductive cloning. And they were seeking the money only to do nuclear transfer of cells.
Which people have no clue what it is.
Exactly, exactly, because they cannot, no one took the time to explain, but it was deceptive on purpose, on paper, and it passed the scrutiny of the state to make it a ballot, and it went on the ballot, and people bought it and voted in favor, and that is the Stem Cell Research Institute in California that is associated with the university system. That was a huge infusion of taxpayer money into the state university system. Back then, it was about 10 years ago, to this day. But the mud in their face is that they have yet to produce a clinical cell line that is viable and doesn't cause tumors. Okay. In the meantime, the adult stuff.
Millions of dollars have been
Exactly.
put into that.
Exactly. And potentially thousands of embryos, right? Because this is a whole state university system. Yeah, so that's what we have. And
But did you see the movie My Sister's Keeper?
no,
It's very
Yeah.
interesting because it's along this line.
yes.
The couple had a child, the
Mhm.
child had leukemia,
Hmm. Hmm.
and then she
Uh-huh.
was used and she was very young to donate her blood to keep her
Sister
alive.
alive, yeah,
And her sister was progressively getting ill,
Right.
and as she grew older, she was tired of being, in the hospitals and always donating marrow and things like that.
right,
So this child was done. Actually, the doctor advised the couple, why don't you have another child?
yes. Now I remember, that was based on a real life situation.
Donate,
Right.
And she went to see an attorney, and the attorney got her case, and she eventually won the case because that was the decision. The parents decided for her, consented for her, but she was a minor,
Right,
so she couldn't.
yes.
She went to
She
the court
could not.
to ask that she would not be used anymore.
Right,
to do
right.
that.
Yes,
And actually, the both sister have spoken, and her sister
Right.
sister asked her, please do that because I am tired.
mm.
She wanted to die, and she was tired of
Oh,
fighting.
look at that, right,
Yeah.
exactly. So yes, it wasn't 100% solution. I know for leukemia, the leukemias are some of the most curable diseases, but it can also be long-term debilitating, and you have to go patient by patient, right? And she was certainly a good match because of the compatibility of the genome and all that. But so the younger sister was being used, obviously.
And she was very little.
Right. Right.
Because she was born for that. Right?
Exactly.
She was created.
She was created for that. She was created for that.
This is have a name. I don't remember, but they say they have a name for them. Babies are made for preserving the life of the other baby.
Sibling.
Yeah.
Right now it has been that way. It was yes. So it was. There was a real case like that. Yes.
For that of the child.
Yeah. Exactly. To know that you're being used as body parts, right?
In the garage.
Exactly. In fact, there was a case, I don't know if it was the same case, but I think I remember for the second child, they needed a girl and she was pregnant with a boy and the boy was aborted until they got the girl. Okay. And there may have been another case when they went through the in vitro thing and they did the sex selection so they would be the same gender and compatibility would be better. So a lot of manipulation of human life, very sad, but it truly is not illegal. I mean, it verges on the horrible, but it's not illegal. So that's when we try to implement these laws that are incremental laws. It doesn't forbid the procedure as such, but to use it, for example, some states are trying to get legislation to prohibit sex selection, abortions or sex selection, even in the in vitro industry, because they're just discriminating on the sex. the child is perfectly normal. All right. But not in some states, it has been stopped because of the federal right to an abortion, even for no reason at all. Emma?
Save your baby.
I'm sorry?
Save your baby or save your sibling.
Oh,
It's
save her.
Google and
Yeah.
Wikipedia, it's a
Yeah.
trend.
Save your baby or save
Save
your
your
sibling.
baby
Wow.
or
Yeah.
save your sibling.
Right. Right. Yeah.
Uh,
Yeah. It's amazing. And some people think actually this is good, right? This is good. You know?
I have a question.
Mhm.
I've seen on videos that they build in the lab like a heart for lungs. What
Yeah.
is that called?
That's different. We're going to see that next week in the following lecture with tissue regeneration and organ regeneration. Even doing now they're doing 3D printing, scaffolding, on which then they pour the cells because the cells need to attach to something. And then that 3D scaffolding is probably something similar to a collagen protein or something that with time disintegrates. And once the tissue forms in a particular shape, I'll show you an ear lobe that was grown on a mouse, for example, the Visconti ear. Yeah, that's for next week. The stuff that it could be good that is going on, that is kind of science fiction, but it's happening already. Hmm? Yeah, you had a comment or
No,
something?
no, I'm thinking.
Yeah.
Because it doesn't make any sense. I mean, these babies are done in vitro, right?
Right,
And they
Yeah.
choose the ones that are compatible. So
exactly.
you're basically killing your other
Siblings,
human
yeah.
babies to save one that is, I mean,
Exactly. It is. It's a very bizarre mentality.
yeah, I mean,
People become possessive and they become, they can do it, they have the money and so forth, and they push it forward. The one case I wanted to talk about last is He Jiankui, because he was in the news just last month. He's the one who claimed to be the first to have done human cloning. So another company up in Massachusetts anyway, it's Advanced Cell Technology, ACT. Advanced Cell Technology is another one of these old biotechs that has been around. And they were doing human cloning. First they were doing cow cloning, because a cow egg you can actually see with the naked eye. So large you can actually just like a little dot, okay? So we're doing cow cloning, and then they started getting a little wild and putting human somatic cells into cow eggs, into nucleated cow eggs, and see how far they can. They were calling that human cloning. It's actually hybridization. But they figured since the genome is totally human, and they just wanted to see if the regulatory proteins worked in the cytoplasm of the cow egg. And they got it to certain stages, but then they would die, thanks be to God. But it was getting very bizarre. At any point, they claim to have been the first ones to have done human cloning through that process, advanced cell technology. But that was in the early 2000s, because Worcester, Massachusetts, because I was in Boston at that time. But anyway, this other fellow, just the last month, is this fellow was giving a talk in a Chinese cell symposium, and he claimed to have been the first one to have edited the human genome. All right, edited the human genome. This is the technique called that thing. CRISPR is the acronym CRISPR for, it's a transfer of a DNA sequence from one species into another species. Now, we have been doing that in the lab for years, all right, gene transfer, but it was pretty much shotgun. You could insert the DNA into the cells of the other species, into the nucleus, but then you'd have to wait and see where the DNA inserted into the genome and where it was expressing, all right, where the protein was expressing and so forth. But now with this technique called CRISPR, that DNA sequence can be tagged and inserted into a specific region of the DNA of the host species. So they can insert it into a particular region, which is going toward the way of actually gene editing, correcting defective gene sequence, whether it's human or non-human. But if there's a defective sequence and I can swap that sequence out and insert the correct sequence, right, then that's a good thing. We're healing at the level of DNA. So that's the gene editing issue, right? He was giving a talk and he claimed that he had done that, but he had done it some time ago, at least a year ago, because the twin girls had been born already. I think it's Nana and Lulu. I think was their name. Let's see. But what he claimed was that he inserted Nana and Lulu babies. So there's already a Wikipedia on it. These are twin girls. They're 3 in vitro, of course. There's CRISPR. So what he did, so he claims, is he got 2 twin zygotes, right, from the same mother, same father. Got wait, I don't know if he did the cloning. He had 2 twin zygotes and to the blastocyst at the zygote level, and inserting some gene editing so that inserted it a sequence that prevents the HIV from coming into the cell. Right? So he made those cells immune to HIV and implanted the embryos. 9 months later, the girls were born. He claims all that. And these girls are immune to HIV. That's what he's claiming. By doing the gene editing at the zygote level, of course, it has to involve in vitro, big time, all right? And trial and error, because who knows if it's taken or not. But the thing is that he can insert that sequence in a specific region of the DNA. And he claimed it, but he hasn't presented the scientific evidence. In the meantime, the university where he was doing the research went ballistic because the university did not know that he was doing this. And there were protocols that he should have followed. He had to go through an IRB and ask permission in the institutional review board to do the experiment, present the protocol and all that. He did nothing of that because he claims that he was on sabbatical and that he did it on his money, and therefore he didn't have to consult anyone. Yeah, but he's using the facilities of the lab and everything else, plus totally unethical. He bypassed all of the controls that even China, as licentious and as liberal as they are on all this, even China was against it. And the university was totally taken aback. They've done a case for him, on him now, and all of a sudden he's disappeared. No one knows where he is now.
He had permission of the parents only?
I presume he did, but I don't know, strictly speaking, I don't know. But he ended up with 2 monozygotic twins where he edited the gene there using CRISPR, and then move it forward, and 9 months later, supposedly these girls are born and they are immune to HIV. So, you know, potentially it has a lot of good, but it has a lot of bad because he's manipulating all these human embryos. He didn't follow any of the protocols, and the scientific community is against him, and everyone in the scientific community has spoken against these experiments, simply because he did not follow protocol, and he did not put the protocols through the institutional review board like he should have and all that. In other words, he didn't have permission. But the bigger question is, had he done that and with very strict protocol, maybe he could have gotten approval for it. and then it would have been, legal. But the question is ethical. That's the whole point. It's unethical because he's doing human cloning without the consent of the victims to do all that and to risk their life and so forth. And so that's why we would be against, in principle, bioethics, we would be against this manipulation. But
and if it works, we can assume that he must have done like 200 trials before.
exactly. On average, there would be 200 other of those girls that didn't make it because of the trial and error procedure. Okay, but he's only telling us about the success because obviously he was looking at the Nobel Prize. But it backfired on him. Now, the reality is that at this point, he's disappeared from the map. Nobody really knows where he is, somewhere in China, maybe. But we're still to see the evidence, the scientific evidence for what he did, the exact protocol, and if it really worked or not. Then what are we going to do? We're going to expose these girls to HIV to see if it worked?
Exactly. That's what I was thinking. How do we improve that?
Exactly. Right, or marry them to a contaminated husband, and it just gets more and more bizarre and totally unethical. See. Yes, of course. You have to leave early. Sure, no problem. Thank you.
I know this technique, Father,
CRISPR.
they use it a lot in for yeast in biotechnology in the review industry to make beer.
Oh yes,
So they,
for yeast, huh? Yeah,
they
specialized.
Manufacture a yeast
Right, that has particular properties. Yes,
So you have the flavor and everything.
exactly. Yeah, so there's a commercial angle to it. Exactly. Yes, yes. Oh, it has great potential, really. But again, we go back to the very beginning of the lecture. Is it human or not human, the research? We're experimenting on plants and animals or experimenting on humans. So certain things that may be done in animals may not be done in humans because of the intrinsic dignity that we have. Simply, we have to stay away from experimenting with people whom we cannot get the consent from. And it's not, we're risking their lives, basically. So, all right. But this gets into also the whole area of GMOs, genetically engineered organisms and so forth, right? Where, for example, on the good side, to be able to insert antibiotics into wheat, for example, or to make them drought resistant and so forth, or fuller grain. So it's artificial selection, but down to the molecular level, which is pretty sophisticated. Anyway, we're going to look at some of the positive stuff next week on fetal surgery, for example, and things like that, which is looking at some positive things that modern technology is able to offer. But this one, because it involves the gross manipulation of human life, either the stem cell research or the cloning, we have to be clear on these distinctions, right? First, on the stem cell, what kind of research we're talking about. At the end of the day, the adult and the umbilical cord seem to have the best promise. And then on the cloning, essentially for human, we have to stay away from it. Okay, I think that's it. We look at the little sheet that I gave you. The two levels of distinction, the cord blood, ISPS, the induced pluripotent, the nuclear swap, and the two types of cloning. Okay, so that's about it.
I
Monica?
forget what I was looking at, but
Yeah.
when I get that light can.
Oh, yes.
A paper that talks about what companies are doing.
Yes, exactly.
I'm not sure if it was in there or somewhere else, but it talked about food companies. And
Right.
I went to this site, and I forget what it was, but there were a few things on there, and one of them was coffee. And I don't know why they do it, but they somehow use embryonic stem cell
For coffee?
in this coffee mate, in this, like, not in
Oh,
the
coffee mate.
but to experiment. I don't know if it's the
Hmm.
flavoring or whatever, but I have no idea how how they got to that
Wow,
conclusion. to
look at
use
that.
embryonic.
Yeah.
But I read that and I thought, I can never
Wow.
drink that again because
Wow.
I know that
What's the
this is
connection, yeah.
involved with.
Yeah. We'd have to, yeah, well, we'd have to see. So Coffee Mate, I think it's polysaturated lipids or something.
Yeah. Yeah.
Right, the creamer.
Isn't that bizarre?
At the molecular.
What does it have to do with embryonic?
Yeah.
But maybe they're going to cows for the milk. Maybe it's not human. Is that human?
Human embryonic? You know, with all due respect to LifeCam, we have to be careful how much they're taking it and how solid their research really is, okay? Because they may see at one point it was circulating with regards to cosmetics that they were using embryonic fetal tissue for cosmetics because it's more compatible with the skin and so forth, and they were just going around the abortion clinics and asking for the fetal remains, right? And using that and grinding it up and using it for mostly the collagen source, collagen. But we have to be careful. I really like to see the research first before we go there. But
Can you search it for next week and you can write it?
yeah, it'd be interesting to see if
I don't actually.
Well, yeah.
yeah, or the next time. Yeah, that would be interesting too. Right. Yeah. That's true. Yeah, well, that would be an interesting thing to research, right? And let's see. Yeah, we do have class next week and the following day. There's one coming up
We don't have class on the 16th.
on the 16th, in two Saturdays. Yeah, because well, I have a prison ministry, but it coincides with the spring break. Happily.
On the 16th, right?
Yes, 16.
That's the only
That's
one.
correct.
And then the
Yes.
next one is in
Right. We go up
May. We
until
only have
the
2 Saturdays off.
yes, that's correct. Yeah.
16th of March?
16th of March.
so the
Yes.
spring break.
Right.
The 20th is on Saturday.
Right. March, May 18. May 18.
May 20. No, is it May?
No, May 18.
And beginning
It
of
may be May 20 is a Monday, but it may be lecture
I'm
number
sorry, it's
20.
April 20. April 20 is on Saturday.
Let's take a look. Yes, you're right. April 20 is Holy Saturday, so no class then. Right, April 20.
Because this course ends on the 4th, but we just start the
Yes.
second semester right after.
Yes, the following weekend, yes, right. Yeah, it'll be a short one because it's only 8 weeks.
So beginning
sorry.
of human life, Father, ends in,
Next class, and then we'll start on end of human life.
so we only have one for this.
One more for this one, yes. So it's 7 and 7. It's a total of 14.
Okay.
Yeah. So next class will be the last one on the beginning of human life issues. And then following, we'll start on end of human life. And we'll have 7 lectures there on the end.
And Father, we should still do, we need to do.
Oh yes, the modules, which, yeah, I've forgotten to come back to the modules. Right, right. Supposedly they're working already, but I haven't gone into it. I need to do that. Yeah. Okay. Yeah, also, while we're talking on the calendar, together with some other colleagues here, I'm organizing a second international conference on climate, nature, and society. It's good as a good primer for the environmental course, environmental bioethics. 3 years ago, we had a first conference here at St. Thomas, based on Laudato Si. Have you ever heard of Laudato Si? Laudato Si is the first encyclical in the history of the Catholic Church. Yeah, Pope Francis wrote it for care for the environment, care for creation, okay? And it was published in the summer of 2015. So in February of 2016, we organized an international conference here on campus on care for creation, on climate change, basically, and global warming. It was a 2-day affair. The first day was on the scientific evidence for climate change, because that's controversial to this day. And the second day, the second day was some of the sociological and spiritual implications of climate change for us and for future generations, like sea level rise, for example, and the flooding that is occurring on Miami Beach and so forth. And that conference 3 years ago was very successful because we had 6 speakers, 3 scientists on the first day, and 2 sociologists, and Cardinal Peter Turkson. Peter Turkson is the cardinal who wrote the draft of the encyclical for Pope Francis. So that was a very high level conference that we had here. We got him. It was last minute, but we got him, and he was a keynote speaker on day 2 to a jam-packed audience there at the auditorium in the School of Law. Anyway, some people who were in the audience was the state representative, the Florida representative of the Nature Conservancy. I don't know if you're familiar with this group, but the Nature Conservancy is an environmental group that is nonpartisan, and they're interested in promoting care for creation, right? They have a very sophisticated website, and they try to lobby in favor of the environment, but also they have projects of restoration of areas that are, especially in the urban areas, to restore them to their natural thing. The other day I went to, so I'm inviting the high schools also, in the Catholic high schools in the area, and I went to Archbishop Carroll High, which is in the West Kendall area of Miami. It's one of the Catholic high schools. And the principal of the school, Dr. Jean, he's from Jamaica. And I told him about the Conference of Forces. Oh yeah, I know the Nature Conservancy, he says. The Nature Conservancy in Jamaica, they restored an area of coastline in the northern part of the island that is near Kingston and some other urban area. They restored part of the natural environment there. And in the southern part of the island, they have a tilapia farm, which is for fish, right? And so these guys are very big, they're international. Well, we didn't know about this, but it turns out that the state representative of the Nature Conservancy was in the audience at that first conference. Then, sometime after the conference, she got in touch with me and she said, we like your conference very much, and if you're interested, we'd like to sponsor you for if you want to organize a second one.
Wow.
Very good.
So it's taken 3 years, but here we are now. In April, we're going to have it, and this coming one is going to be faith-based. All right. So now we have our second international conference on climate, nature, and society. We have a web page. It's very simple. You just Google STU Climate Conference. This STU Climate Conference, and we are there, the first link. It's our website. And so it's coming up April 4 and 5th, the first Thursday and Friday of April. And we have the schedule of events, 2 days. We have about 16 speakers, including the Archbishop. Archbishop Wensky is giving a talk. From all different walks, we have Protestant, we have Jewish, we have Muslim, even secular. We have people from the county, from Miami Beach, from Dade County Urban Planning. So these are all the speakers. And at the very bottom, that's Cardinal Turkson there. This is a link to all the talks of the first conference. So this is a very complete website here. Okay, we have all the talks. And actually, the first one starts with Dr. Muller.
Thank you very much. And it's a delight to be here. Let me get started.
Yeah.
Take a look at the background picture there.
That's Dr. Muller, Richard Muller. He's an astrophysicist at Berkeley, University of Berkeley, and he has a center there called Berkeley Earth, and it's about the environment. What's interesting about him is to in his own words, he's a converted skeptic. He was a skeptic with regards to climate change because of all the political spin that has become politicized and partisan politics. So he was very skeptic, but he wanted to see the hardcore evidence. Being an astrophysicist, he saw the evidence, and now he is an advocate of.
That's a typical morning in Beijing.
For example.
The pollution is horrific.
Yeah.
What does that have to do with climate change? I hope to show you that they are very closely linked. And in fact, since they go together, if we can address this, I think it's the best way to stop global warming. That's the bottom line. That's the straight.
Et cetera, et cetera. So it's 2 days of conferences, okay? But if you're interested, you can take a look. Each talk is an average of about 45 minutes to an hour. And, yes, absolutely. Yeah, and it's free of charge. You register, you register here. Just this button, right? You go in there.
Would it count in an hour's file?
Yeah, you can put it, as a matter of fact, you want to put it for, why not? Might as well get something out of it. You're always looking for you're looking for internship hours. Sure, why not? Absolutely, yeah. Because, and then the idea is to try to engage the speakers in their thing, because this one is faith-based. So the idea is, what are different faith communities doing to care for creation? And so we're looking at the local level, at the regional, national, and international level. And we even have a speaker from the Vatican, Tebaldo Vinciguerra. He's coming from the Pontifical Council for Integral Human Development. All right, and he's a spokesman for the Vatican. He's an expert on water, all different aspects of water. So he's part of it. And in fact, the president of that council, that Pontifical Council, is Cardinal Turkson. So Cardinal Turkson is his boss, and he's coming officially representing the Vatican. We're trying to get a little statement from the Pope for the conference. Let's see if it works. But I think it would be neat to show the Catholic perspective. What are Catholics doing for, got a map where we are and everything. So just register, your convenience. You go in there. And it's just basically name and how many people you're bringing with you. And then he'll ask you for your name and email address, basically. Oh yeah, we have for each day. You have to register for each day, because some people may come day 1 and not day 2, etc. All right, and then the email. That's it. So anyway, I'll tell you more about it as we get closer, but that's basically the second conference coming up. Yeah, I mentioned that because it's that April 4th and 5th is just before. We have a class on April 6th. So it's right after the conference. Okay.
How do you keep track of the hours of the internship?
Yeah.
How do we?
I had sent an email early on, but I don't know if you remember it. But so it's 2 things. One is the email. Let me see. Let me just go here for a moment. I can send it again, but basically, let me see. Here. Internship. We're 3rd. Yeah.
How do we keep track of?
There is, he sent,
I have it.
This one here.
We have to write in this.
just the date and the hours. that you-
Exactly. I'll show you because Gemma.
I don't even.
Yeah, not even.
That's where.
Right, just the day and the hour. So this is Shema's, for example, okay, because she did it, the internship that she did with one of the politicians. And so just the date and the hours, and it's easy because once you add that up, right, to 150, that's it, you're done. Okay, now you can do it in different places, different venues. If you want to put more information, that's fine. I'm not requiring it. In other words, this is on the honors principle. I trust that you give me the true numbers,
Of course.
and that's it. If you want to keep it for your own record, you can expand here and you can put where you did it or some contact information if you later want to go back there, whatever. You can do it several different places, like I said, if you want to do a variety. In fact, Hema did this early on simply because she wanted to, and she got that internship with the politician. But now she's going to go to Respect Life office here, and it's beyond this, but she continues, you know.
And when we finish, you send it to me.
Yes, you just send me this.
150, right?
Right.
For November.
Yeah, exactly. You have up until the end of November, which is the drop dead bottom line, because I have to enter a grade for it, which is, by the way, it's just pass-fail, this course, the internship. So that's one. And the other one is the actual form. Let me show that to you here. Oops. Disappeared. I have to rescue it somewhere. Okay. I'll rescue it. I have it somewhere else. Maybe, oh, okay.
So the
I don't know where to
conference
find
will
it.
be 8 hours in the first one.
Yeah, yeah, there's a lot of hours there.
No, it's interesting because you know people.
Exactly, yeah, you can network and synergize. Here's the form. So statement in the front, the beginning. This is for the supervisor. These 4 questions. It's mostly about, you know, professional ethics, if you're showing up on time, if you're learning something from it, and so forth. And then one final question, any comment that the supervisor wants to give me confidential. Typically, the comments are very good, you know, we're very pleased to have this person, of course, for free and everything else. And then they can just, once they fill this out, see, it only needs your name, not the signature, because the student is not supposed to see this. The supervisor will give you feedback, right? But since it has a confidential question there, they just put your name and then their signature.
And we need only one because if we do
Only
a
one.
different.
Only one,
Okay.
just one, because this is for compliance. In other words, so that I have something on record to justify the passing grade.
Okay.
You know, it's a compliance thing. If it were up to me, you don't even have to do this, but
Yeah,
I need some kind of a paper trail for in case we get audited or something. I don't know. Okay, so that's
Yeah,
That's
like
the
for
fun.
me, I do all the CME, the continuing medical education, so
There
I
you
have
go.
my certificate.
exactly.
Because I have, those serve me for my licensure.
Yes, you have to present a certificate or something, right? To maintain the license.
For my boards, you know,
Exactly.
medical education.
Yeah, and that's on a yearly basis, right?
Yeah, every 2 years.
Oh,
For
2
like
years.
the license.
Oh,
And then
okay.
for my boards, it's every 10 years, I have to have certain number of hours.
Oh, okay, okay.
Like 200.
Okay, so 2 and 10, yeah. Yeah, for those talks, for those hours, the people, the speakers need to be qualified. Typically, you need a terminal degree. It doesn't have to be an MD, it can be a PhD, because I've even talks for CME credits.
Yeah.
But they have to have terminal degree and so forth, and they have to present like a little syllabus and objectives. Right, so handout with objectives and how the objectives are assessed. So it's technical and it's, you know, it's pretty professional. Okay, this one is a simpler version of that. Okay, excellent. I don't have anything else. So thank you once again. And Rizal, thank you. We're going to log out now and I'll send you the link as soon as I have it, okay?